Hello, my name is Pugmire, and I will be giving a tutorial on how to stain a cheek cell. The materials you will need for this experiment are the following: a slide, a cover slip, water, dropper, toothpick, microscope, methylene blue dye. The slide is where you will be placing the cells, and the cover slip is placed over top it for a better viewing experience. The water, you don't need very much of it. In fact, you only need one little drop. A dropper will help you do that. The toothpick is what you'll be using to collect the cells. The microscope will allow you to magnify and see the cells more clearly. And the methylene blue dye also aids in the viewing experience. To begin, you will take the slide and place a small drop of water on the clean slide as viewable here. You will then take the toothpick and rub it around on the inside of your cheek. You may not believe it, but this collects hundreds to even thousands of cells. Then take the toothpick and rub it around gently on the drop of water, as the cells will be collected and then displaced into the water. You should be very precautious while using methylene blue dye. It can stain clothing and skin for a very long time. So you should be very careful while taking it out of the bottle, and while using the dropper, place a very small drop of the dye onto the small drop of water, as seen right here. This next step is very important that you do it right, yet it is very tricky. You must angle the cover slip onto the blue dye and press it down firmly so all the air bubbles are gone, as seen in this picture. You must make sure the angle is correct as you place it down, otherwise air bubbles will be there. The next slide shows a video of how to do this properly. The dye is very strong, and if there's too much of it underneath the cover slide, it will cover up the cells and not allow you to view it. So you must draw the dye out by using a paper towel and going around the corners of the cover slide and pulling the ink out. And there's a video in the next slide that will show you how I did this. After pulling most of the dye through, your slide should look like this. As you can see, it is not the dark blue like the dye is, but it is a very tainted light blue. This is perfect for microscope viewing. Make sure that the microscope is on the lowest magnification, then carefully place the slide under the lens and using the metal clips to make sure the slide stays in place. For the next part of this experiment, it is important that you know the difference between the coarse focus knob and the fine focus knob. And what they both do is that they move this platform up or down. And this movement causes the specimen, or the cells you are viewing, to become more blurry or less blurry. And this focus can determine the difference between a good viewing experience and a bad viewing experience. The coarse focus knob is abrupt change in the platform direction. It is a very fast movement, and this can be useful for moving the platform down or moving it up closer to the lens. But the fine focus knob should really be what you use the most, as it carefully brings the measurement that you need for your viewing experience. If you were to try to use the coarse focus knob the entire time, the picture would most likely be blurry, as you cannot focus and fine-tune it, and that is what the fine focus is for. Using the coarse focus knob, bring the cells closer to the lens, then use the fine focus knob to clear and focus the image until you can see the cells perfectly. The picture you see in your microscope should look a lot like this. Then lower the platform down and then switch to the medium magnification. If you do not use the coarse focus knob and lower the plate, then there's a chance that the medium magnification might slam into the plate while you're trying to switch it. this happens, you could break the magnification and it is very expensive. So make sure you take all precautions necessary and just lower the plate all the way down. It may take more time to bring it back up, but it really in the long run, it is important that you bring it down so the rest of the instruments are safe. Repeat the steps as for the small magnification and bring in the cells to focus. When you're done, the cells should look a lot like this. Repeat the steps as before, lower the plate and switch to the high magnification. This will take more time as the focus is harder to get, but the results are astounding. When you're done, the focus should look a lot like this. I've taken the privilege to zoom in on this picture and enhance it for better viewing in the next slide. After zooming in on the picture and enhancing it, this is what I arrived at. Some of the key features for this photo that you should keep in mind are the cell membranes and the nucleuses on the inside of the cells. It is really cool that we were able to see that. The next stage for this experiment is the cleanup. Make sure you carefully remove the slide by taking off the clips, and then slide the cover slip off the slide very carefully, and afterwards rinse and dry off the glass. Gently unplug the microscope, wrap the cord up, and place the cover over the top of the microscope. Then place the microscope where your teacher directs. The key ingredient for science is our ability to share. So make sure you share your findings and explain the process of how you came to these results. Thanks for watching this tutorial. I hope this helps you complete your staining of your own cheek cells too.